Hello everyone, good afternoon. I'm sure you are aware of the recent drug recalls due to nitrosamine impurities. As we know, nitrosamines are mutagenic and carcinogenic, and these need to be controlled at a very low level. In fact, any nitrosamine for which no toxicological data are available needs to be controlled at or below 18 nanogram. That is, the acceptable daily intake is only 18 nanogram. Now, nitrosamine impurities can be present both in drug substance and in drug product. In drug substance, these impurities come from manufacturing processes, but in drug product, it can form during manufacturing and also on storage. It is possible to control nitrosamine impurities in drug substance by modifying process steps, for example, implementing different synthetic routes, effective purging, etc. But when nitrosamine forms in drug product, it cannot be purged. So the best way to mitigate this risk is to stop the formation at the first place. Our research has demonstrated that it may be feasible to inhibit nitrosamine formation in drug product, both in solid dosage form and in solution. Let's look at the chemistry of nitrosamine formation, although from a very high level. We need two ingredients to form nitrosamine, nitrite and a vulnerable amine. And in this case, I am showing a secondary amine. Nitrites, when react with acid, it forms the active nitrosetting agent NO+, or for that matter, N2O3. And this active nitrosetting agent reacts with the secondary amine or any vulnerable amine to form a nitrosamine. We should remember that the optimal pH to form nitrosamine is around pH 3. So at that pH, nitrite can react very efficiently with a secondary amine to form nitrosamine. But nitrite can also directly react with secondary amines in the presence of aldehyde at neutral or at higher pH. Also, tertiary amines can react with nitrosetting agents to form nitrosamine, although the rate of nitrosamine formation is much slower than that with secondary amines. Another thing we should remember that nitrate, not nitrite, nitrates are not a problem for us. Usually, if we have nitrate as impurity in drug product, they will not form nitrosamines under the conditions which drug products usually experience. So in summary, we need two ingredients to form nitrosamines, mainly nitrite and a vulnerable amine, which is most of the time a secondary amine. And when that happens, when these two ingredients are present in drug product, nitrosamines can form, and in drug product, we cannot purge nitrosamines when it forms, so we need to inhibit it. Now, how can we inhibit it? What are the mechanistic pathways? In our research, what we did, we exploited two modes. The first mode is consumption of active nitrosetting agent, and the second mode is consumption of nitrite. Under this first mode of consumption of active nitrosetting agent, there are two mechanistic pathways we exploited, which is the first one is a redox pathway, and the second one is diazotization. In this case, for the redox pathway, the inhibitor I have shown here is ascorbic acid. And for the second pathway, the diazotization pathway, we used inhibitors where it contains a pre-primary amine group. And for the second mode of inhibition, the consumption of nitrite, we used polyphenolic inhibitors, which react directly with nitrites to consume it. to show the feasibility of inhibition of nitrosamine formation in solid dosage forms, this amine, 4-phenylpiperidine hydrochloride, was chosen as the model drug. And these are the inhibitors we used: ascorbic acid, sodium ascorbate, alpha tocopherol, ferulic, and caffeic acids. Now, what we did, we used this amine as model drug and made tablets, 100 milligram tablets. And we made three types of 100 milligram tablets. The first type, there was no inhibitor was spiked in the first type of tablet. In the second type of tablet, we had 1% inhibitor spike, 1% by weight, about 1% by weight. And in the third type, we had 0.1% inhibitor spike. And to make these tablets, we used these common excipients. Now, from your previous talk, you know that these excipients, they contain nitrite as impurity. That means in our tablet, we had a vulnerable amine, 4-phenylpiperidine hydrochloride, and nitrite from these excipients. That means we had both the ingredients to form nitrosamines in drug product. What we did then, after making these tablets, we subjected them to accelerated stress at 50 C, 75% RH. Although this is a very harsh condition, but we didn't want to wait long time. So we wanted to stress them very high, very harsh, and see whether our inhibition mechanisms really work. Here is the data. After stressing the tablets for 1 month at 50 C, 75% RH, the tablets containing no inhibitor formed 345 parts per billion nitrosamine in 1 month. But the tablets containing all these 5 inhibitors, they form nitrosamines in a much, much lower level. In fact, if we calculate the inhibition efficiency of these inhibitors, we can find that all of these inhibitors inhibited nitrosamine formation in greater than 80% level when they are spiked at 5.7 micromole level. That means if we think of ascorbic acid, it is 1% spike. This is quite remarkable. With this inhibitory efficiency, we can basically achieve the goal of acceptable daily intake of a nitrosamine 18 nanogram or below, no problem. And what you saw that, what you calculated, we will achieve this goal with most, if not all, drug product. Now, this is what I showed here. This is the inhibition in solid dosage form. But what happens in solutions or suspension, because we market drugs in solution and suspension too. For that demonstration, what we did, we used again the same drug, same amine, and this is the reaction condition we used: pH 3, 60 degrees Celsius, 23 hour. This is a very optimal reaction condition to form nitrosamine. And in this case, what we used as inhibitor are these 3 amino acids: glycine, lysine, and histidine. And all these 3 amino acids, being amino acids, they had free NH2 primary amine group present in them. And we know from my previous slide that when free amino group is present, the inhibition mechanistic pathway goes through diazotization. And what I found that all these 3 amino acids, they did a pretty good job in inhibiting nitrosamine formation. Glycine at 44% level, lysine did it almost 57%, and histidine inhibited nitrosamine formation at greater than 90% level. That means, in theory, we should be able to inhibit nitrosamine formation in solution or in suspension drug products too, if we use proper inhibitor to do the job. In summary, what we have shown here, inhibition of nitrosamine formation can be achieved via multiple mechanistic pathways. Our model study demonstrates that inhibition of nitrosamine is possible both in solid and in solution drug products. We got to remember that we have shown only 3 mechanistic pathways, but there could be more pathways available. For that, more research will be needed. At the end, I would like to acknowledge the contribution of my colleagues. Without that, this project wouldn't have been successful. And I thank you for your attention.